am Heidi Lovig with Marine Applied Research and Exploration, and I'm here today to present the North Coast mid-depth and deep subtidal ecosystem survey results performed using the ROV Beagle shown here. So the overall goal of the project was to describe the baseline condition of 3 distinctive ecosystems in the North Coast within 4 MPAs and their respective reference areas. The 4 MPAs we surveyed included Point St. George SMCA, Redding Rock SMR, Mattole Canyon SMR, and 10 Mile SMR. The 3 ecosystems we surveyed were mid-depth rock, soft bottom, and deep ecosystems or canyons. We used multi-beam mapping imagery to select reference areas adjacent to each of these MPAs to survey that has similar habitats and depths. In order to survey each site, transects were selected within each MPA and its corresponding reference area. This is an example of Redding Rock transect selection shown here, with rocky reef areas of the map shown in brown and the surrounding soft bottom habitat shown in blue. Now within each MPA and its reference area, 2 types of rocky reef transects were selected to survey. The first were in index sites within fixed 500 by 1,000 meter plots. And the second type of transect were transects that were randomly chosen and placed around the reef. And then also within each MPA and reference area, characterization transects were randomly chosen to survey within the soft bottom habitat. So during the first 2 monitoring years following MPA implementation, which were in 2014 and 2015, the ROV and boat surveyed pre-planned transects along the seafloor to collect video and still imagery using 2 cameras, 1 forward and 1 downward facing camera. The ROV pilot and boat skipper navigated the transects using real-time positions of the ROV provided by the acoustic tracking system seen here, and then video and sensor data was transmitted back up the umbilical and recorded back on board the ship. So following data collection, video was brought back to our lab and post-processed, where both forward and downward facing camera views were simultaneously watched to record multiple layers of data. The first of which were substrate types, which were then characterized as either soft, mixed, or hard habitats based on their substrate compositions. Next, the video was watched to enumerate macro-invertebrates. And finally, the video was watched to enumerate and size fish. Now this layered approach to processing video was very beneficial, as there was flexibility to choose which data combinations you'd want to use for different types of analysis. And also additional layers could be added, such as rugosity, which is a measure of substrate complexity, or biogenic habitats, which are habitats formed by the growth of particular species such as seagrasses or kelp. In total, within the North Coast study region, we performed 60 dives surveying over 106 kilometers of seafloor habitats, which produced 97 hours of video and over 16,500 photos, which when analyzed resulted in 92 species and species groupings of fish combined, and 99 species and species groupings of macro-invertebrates combined. So our surveys produced a lot of data, so I'm just going to give you a brief idea of what the baseline data can be used for as a benchmark for future comparisons to monitor changes over time within these areas. I will show you some of our results for fish, abundance, analysis of index site pairs, size, and depth distribution, macro-invertebrate abundance, and some of the initial changes we saw between the 2014 and 2015 survey years. To show fish abundance across the entire region for both rocky reef and soft bottom transects, we grouped fish into 7 subgroupings, the first of which were rockfish, which included larger benthic and epibenthic rockfish species, of which the blue, olive yellowtail, and canary seen here were the most common across all sites. And in total, rockfish as a grouping represented 14% of all of our fish observations within the region. The second grouping were small schooling rockfish, which included half-banded, short-belly, pygmy, and other dwarf rockfish species, which together represented another 14% of our observations. The third grouping were yoi, or young of year rockfish, which were only 10 centimeter or smaller rockfish, which represented 60% of our observations. The next 2 groupings were lingcod and kelp greenling, and they both represented a relatively small fraction of our observations. Next were flatfish, which included any flatfish we surveyed, like soles, turbots, and sand dabs, and combined those represented 4% of our observations. And then the all other fish grouping included species such as smelt, seen here, and others such as eels, rays, combfish, and eel pouts, and combined those represented less than 5% of our observations. Now, of all of the fish species that we observed, only 10 of them were selected for comparison for in-depth density analysis to establish baseline metrics for future comparisons between each MPA and reference area. You can see here I have each of our 4 MPAs in their respective reference areas. The MPAs are shown in either blue or red depending upon their designation, and the reference areas are shown in gray. And for 3 of the sites, we didn't, overall, there wasn't much of a difference between rockfish densities, with the exception of Point St. George here, where rockfish densities were significantly lower in the reference areas than in the MPAs. So we also looked at fish size distribution, similar to what Jan just showed you from Reef Check. We looked at all 4 of our sites, north to south. You see here Point St. George in the north to 10 Mile in the south. If we add a line in here that shows the size at which 50% of the females of the population are sexually mature, you can see for species like yellow eye and canary shown here that the majority of the populations that we surveyed were sexually immature size classes. For other species that were commonly seen in our surveys, such as vermilion and quillback, the opposite was true, where we saw more sexually mature size classes. And yet other species like lingcod showed a geographical split, where the northern 2 study locations had more larger size classes, and the southern 2 study locations had more smaller size classes. We also looked at the depth distribution of fishes in rocky reef and canyon transects. This is just one example of Mattole Canyon MPA, showing the density of select fish species per depth stratified bin. Since this was the canyon, goes all the way from 50 to almost 450 meters in depth. And we noticed that the break shown here between the canyon and the shelf appeared to be an important habitat feature for at least some of the species presented. For example, canary and olive yellowtail rockfishes were commonly found on the flats and tended to disappear around the shelf break. Moving on, you can see yellow eye were only observed near the shelf canyon break. Also, as you would expect, greenling, a shallow water species, were only found up on the flats. And thorny heads, which were a deep water species, were exclusively found in the deeper areas of the canyon along with some of the flatfish species. Moving on from fish to invertebrates, of the over 124,000 invertebrates enumerated, Only 8 of the species groupings represented 90% of all of our observations. The first grouping were slipper and California sea cucumbers, both shown here, which combined represented 40% of our observations. The next grouping were was the white-plumed anemone on its own, and that represented 27% of all observations. The third grouping were 2 rocky reef coral species, the California hydrocoral and short-red gorgonians, which combined represented 10% of our observations. The next grouping were all sea stars combined, which represented 8% of our observations. And then lastly, 2 soft-bottom coral species, sea pens and sea whips, represented 3% of our observations. Now, we did notice few initial changes between the 2014 and 2015 survey years, as this was baseline data. But the most notable change we did see was that 8 of our species groupings of sea stars showed significant decreases in their mean densities between the 2 survey years. However, as you've already heard today, this trend was expected as active signs of sea star wasting disease were noted in both survey years. For example, in 2014, these 3 star species combined, which were the Stimson sun star, sunflower star, and short spine sea stars, we saw approximately 230 observations of those 3 species combined. When we went back to the same sites in 2015, we only saw 16 individuals total of those 3 species combined. And we have plenty of pictures like this here showing them actively wasting. So I'm going to close today by sharing with you some of our video highlights. If I can get it to work. There we go. Of our surveys within the region. We'll begin with the top side view of our deck crew lowering the ROV into the water. This is robotics are a little bit different than what other people use for surveying, so we thought you might want to see it actually happening. So 2 people will sit in the control booth where the ROV is manipulated from the surface, shown there. Oops, I went too fast, so I'm gonna have to go back. Sorry guys. It's gonna be like a minute over. Okay, one more time. You're good. So they're loading the ROV in. There's actually a big box that sits on the top of the research vessel. It's like a blacked out box. And we do most of our manipulation of the ROV from there. And you can see them doing that there. Also GIS, navigational data is recorded in that box. And then within the rocky reef habitats we surveyed, we saw many rockfish species, like the Chinas and blue rockfish seen here. Also, yellow eye were very common in our surveys, with almost about 100 seen at Point St. George MPA alone. And the blues and olive yellowtails, were 2 of our more common schooling species. Also common were short bellies. And then some of our uncommon schooling events included this ratfish grouping. You can see a feather star swimming there, which is actually an invertebrate. Or this group of 5 mola mola we saw swimming together. You usually see them individually, so that was interesting. And then quite unique was this 6-gill shark we saw swimming off of Redding Rock. It's only one of 3 observations we'd seen in the last 16 years. And then my personal favorite, we saw this giant Pacific octopus we caught on film during a feeding event. And then here's our crew retrieving the ROV at the end of the day. See, it takes kind of a lot of manhandling to get that thing back up on the research vessel. And we look forward to sharing more of these types of results with you as we move forward. Are there any questions?
You were fine on time. There's plenty of time for questions.
Yeah.
So, have you ever checked for like bias for some size classes that might avoid the equipment? Do you ever kind of go down there and check to see whether or not there's things you're scaring away that you wouldn't see?
I have to say off the bat that I'm subbing for our main PI who is out today collecting additional data. So I'm relatively new to the project, so some of those questions might be better answered in further detail with him. But I have noticed, we have noticed in general that a lot of fish species tend to not have that big of a problem with the ROV. Like you saw the 6 gill shark, it came right up to the ROV, and other more shy rockfish species that people normally consider shy seem to not have a problem with the ROV. Used to process video before this, and we noticed that as a general trend it didn't seem to be that much of a problem. But in terms of size classes, I don't really know. We tend to not see a lot of really smaller rockfish anyway. Those are difficult to see in surveys in general. But
Great data, great presentation, thank you.
any other questions?
Yeah. Did you guys notice any differences in observations of sea star, the level of sea star wasting between the depths, like as you get deeper?
I don't know. I do know that our Point St. George site, which was one of our more shallow sites, we saw more significant decreases in sea star wasting disease. And I'm not sure if that really answered your question, but I mean, overall, really, it was almost every species that we saw in 2015 was almost completely obliterated from our 2014 to our 2015 survey years. So I'm not sure that depth was much of a factor. I think it was an overall problem.
This might be a naive ROV question, but are there any instruments collecting like temperature or salinity or dissolve, any of those environmental abiotic factors?
Yes.
My question would be more than depth if those actually correlate.
Yeah, we do. We collect. So as the ROV is moving along, all the data that's collected is coded by second, and basically we have, I believe, Hugo can help me with that one. Dissolve oxygen probes.
ROV, so we collect all the CPD stuff, and unfortunately we don't have a pH sensor. So other than that, like, we have O2 sensor and depth and temperature.
So was there a real temperature in that? Was there a change in temperature associated with the wasting?
We're collecting data, but we didn't really look at the temperature yet. So
Okay.
we just probably will.
All right, I might move us along. Thank you, Heidi. Any